Would you like to introduce your student?
Yes.
All right. Well, it's my pleasure to introduce Shelby Wolfram. She's been working with me for about 2 years now. She has been working on a fairly large master's project, of which this is only a small part of, which actually came out as a project actually last fall. So this is all work done probably last half of last fall when all this was done. So,
Literally 2 weeks.
So I will let Shelby tell you about how she looked at these time and duration exposures.
Hello everyone, good morning. So my experiments had to deal with developmental effects of the time and duration of exposure on Xenopus laevis embryos by sodium selenate. So to introduce this, about 1/2 of the frog species in North America rely on ephemeral wetlands to reproduce. Amphibians have a key role in any ecosystem as an indicator species, which just means they're able to absorb pollutants through their skin, gills, and digestive tracts in any area they're living in. So we're going to talk a little bit about sodium selenate. Sodium selenate is a rare element that you find in nature. It exists in inorganic and organic forms. It also has a toxic element to natural ecosystems that are due to bioaccumulation potential. Has a relatively narrow range between deficiency and toxicity, and excess selenium can cause a variety of adverse effects in aquatic organisms. Being used as fertilizer, pesticides, fungicides, really anything that you would use as a nutrient for your plants or to kill off insects. So African clawed frogs is the animal I used for my experiment. They are low maintenance, very easy to take care of. They produce large, robust clutches of eggs, and their embryos are also transparent, allowing us to look inside their bodies to see what's going on. And they do have quick development, so our experiments last about 96 hours, which is the perfect range for these guys. So why am I doing this experiment? Well, fun fact. When I was doing my experiments for my actual thesis, my highest concentration for the selenium was 100 milligrams per liter. And throughout that experiment, I found out that all of my embryos that were exposed to that highest concentration were looking perfectly normal. They didn't look like they were going to die, which made me terrified that my experiments were going to fail. On 96 hours, they all died. They looked horrible. There was no way that you would even imagine they were looking perfectly normal for the first 72 hours. So we wanted to figure out why that was happening. So in our case, we wanted to see if the time of exposure was more important than the duration of exposure. So for my hypothesis, I said that the embryos in the developmental stages has a higher impact than the duration exposed to sodium selenate. So we're going to see if that's the case. And the objective was to determine if the time exposure of the duration of the exposure is more important to the development of Xenopus laevis embryos to the developmental toxicity of sodium selenate. Material and methods. So first thing we got to do is we got to get these guys breeding. So we inject them with a chemical called human chorionic gonadotrophin, allowing them to induce breeding. And we put them in a false bottom chamber for about 12 hours so they can breed. Once we notice that they're bred and successfully laid fertilized eggs, we take them out and we L-cysteine those eggs to get that jelly coat off of them. Once we do that, we make sure that the eggs we're using are blastula to early gastrula stage, and we do a double sort to make sure the eggs that we're using are good eggs. We use 60 to 15 millimeter petri dishes, and each petri dish has about 20 embryos. We keep those embryos in an incubator that's about 24 Celsius, give or take 1. And every day of the experiment, we check for mortality, check the pHs, and we change solutions daily. On the last day, we have to put MS-222 in their solutions to anesthetize them so they don't move when we look for malformations and do length measurements. And so the endpoint is we're looking for mortalities, malformations, and the length measurements for image J. And the experimental design, each experiment that I did, which was 3 of them, was 640 eggs in each experiment, which was about 1,920 embryos. That was a lot. Each treatment that we did had about 80 embryos, so 20 embryos per dish. And if they were not an actual treatment, they were in a VTAC solution. And if they weren't treated yet with the sodium selenate, they would stay in VTAC until they were treated. And they were in 100 milligrams per liter of sodium selenate because we knew that we were going to see death at that concentration. And then we have our timed exposures here, so 24 hours, which is day 0 to 1, 1 to 2, 2 to 3, and 3 to 4. And then we have duration exposures, which are just periodically they're being exposed to sodium selenate, which is 96 hours, 72 hours, and 48 hours. And as you can see from this picture, these embryos, they actually don't look very bad. We had a very hard time deciding whether or not they were going to be dead or not, because we had to look for a heartbeat. These guys, they look dead. There was no denying that they were dead. So our first concentration response graph here, we have exposure time on the vertical axis, and then we have response on the horizontal. And this is our mortality graph. So this is showing us the death in each concentration or each day. And as you can see here with day 2 to 3 and 3 to 4 on our timed exposures, there was significant difference between our controls. That's what these asterisks here are for. And then all of our durations here, they all had 100% mortality in each one. And then we can correlate the colors. So if it's a blue, that's from test 1. Red is for test 2. And green is for test 3. This is our malformations concentration response graph. And we saw only a significant difference with the 2 to 3 and 3 to 4 days. Unfortunately, we only saw one test from test 1 on day 1 to 2 had a significant difference. Not really sure what could have occurred there, but it was interesting to see we had one test that had a significant difference there. And then for our concentration growth graph, we had significant difference with days 2 to 3 and 3 to 4. And days 0 to 1 and 1 to 2, we really didn't see much significant difference with any of our tests. These were some malformations that we saw throughout our test. Here is our control or healthy embryos. We have embryos that had edemas, hemorrhages, bent spines, facial abnormalities, small eyes, the guts are completely destroyed, and then these horrifically severe abnormalities. So conclusion, embryos exposed to sodium selenate continuously for 48 hours or more all perished at 96 hours. Earlier developmental stages were impacted minimally, and later stages that were exposed for a period of time showed devastating effects in the malformation, and they would intentionally lead to their death. So discussion, when we did this, we figured out that the ones that were not dying from day 0 to 1 and 1 to 2, they probably did not have a receptor that had developed yet, not allowing them to be affected by the sodium selenate yet. But once they reached the 2 to 3 to 3 to 4 stage, then they had that receptor that developed that was causing them to get affected from sodium selenate. Future studies, in order to determine if it was only affecting between those 2 days, we need to do longer experiments and do longer time, longer days, so like we can do exposure times from, instead of all the way up to 3 to 4, 4 to 5, 5 to 6, so on. And that will help us determine if it really is a time exposure issue. So I would like to acknowledge the biology department for their resources and Dr. Rayburn for persuading me to do this experiment, even though he knew I didn't want to. But at least I got my answers answered. Thank you very much, everyone. Any questions?
I have a question.
Yes.
Is there, would you ever expect to find that concentration of the sodium selenate in a natural runoff situation?
From what I've read in my research, no.
Okay.
It's been less than 1 milligrams per liter in the natural environment.
And not that that's not bad, I just wondered if there was something, a precipitating event that led you to say, okay, this is what's happening here and this is the result.
Mm-hmm. Yeah, we went this high with our concentrations from previous experiments that I found in my literature of research.
Thank you. Well done.
Any other questions?
In your, where they all died, the time exposure where they died, they all died on the last day?
In our timed exposures with 2 to 3 and 3 to 4, they did all die.
But on,
Or
the,
no.
last, but on the last day?
On the last night, no, they did not all die.
Okay.
So on the last day, it was difficult determining which ones were dead or alive because they had such a faint heartbeat. We literally had to look at them under a microscope for like 5 minutes to find the heartbeat. It was horrific.
I gotta say, this is not a question, but a statement. I've seen a lot of things on Xenopus laevis, and I did not know what they were even talking about at the beginning. You did a really good job of explaining some of the things that I wish I had seen the first time I watched Xenopus laevis with something experiment.
Agreed.
Appreciate that.
Any other questions for our speaker?
Thank you very much.